Hello students, today let us study about totipotency and morphogenesis. First, let us start with the concept of cellular totipotency. The capacity or the inherent potential of a cell to give rise to a whole organism is known as cellular totipotency. This phenomenon of totipotency is an amazing developmental plasticity that sets plant cells apart from most of their animal counterparts. Plant cells retain this capacity even after a cell has undergone final differentiation in the plant body and have the ability to initiate cell division from almost any tissue to regenerate in response to particular stimuli, as long as they have an intact membrane system and a viable nucleus. When plant cells and tissues are cultured in vitro, they generally exhibit a very high degree of plasticity, which allows one type of tissue or organ to be initiated from another type, and subsequently whole plants can be regenerated. This regeneration of whole plants from plant cell culture, when given the correct stimuli, provides the most compelling evidence for totipotency. Expression of totipotency depends on competence or the endogenous potential of a given cell or tissue to develop in a particular developmental pathway, and determination in which cells become irreversibly committed to a particular developmental pathway. For a differentiated cell to express its totipotency, it first undergoes de-differentiation, followed by redifferentiation and regeneration. De-differentiation is the phenomenon of a mature cell reverting to the meristematic state and forming undifferentiated callus tissue, or an unorganized mass of loosely arranged parenchymatous tissue, which develops from parent cells due to proliferation of cells, while the phenomenon of conversion of cells of callus tissue to whole plant or plant organs is called redifferentiation. The genesis of an entire plant from cultured explants directly or via callus indirectly is called regeneration. Let us now study about differentiation. Differentiation is the development of cells or tissues with the specific function and the regeneration of organs or organ-like structures or proembryos like organized structures, usually from undifferentiated tissue. In plant cells expressing cellular totipotency, regeneration of organs occurs not only from undifferentiated tissue but also from previously specialized cells that would not normally give rise to organized multicellular growth, example the epidermal cells, pollen grains, etc. Generally, differentiation may be categorized into two groups: structural differentiation and physiological differentiation. First, let us see what structural differentiation is. Structural differentiation is further distinguished into external and internal differentiation. External differentiation leads to the development of external structures, and the most common examples are root and shoot differentiation, differentiation of vegetative and reproductive phases of life cycle, and the differentiation of reproductive organs resulting in the development of male and female organs. While internal differentiation includes differentiation of various types of cells and tissues. The differentiated cells mostly occur in groups forming different types of tissues, for example, the vascular tissues. The second one is physiological differentiation. It leads to the development of different physiological processes. For example, the variations in the structure between root and shoot are the expressions of different fundamental physiological processes in the two structures. Next, let us see cyto-differentiation. The cells in a callus are parenchymatous in nature, and the differentiation of these cells into a variety of cells like vascular tissue consisting of xylem and phloem is required during redifferentiation of the cells to develop into whole plants. This redifferentiation of cells, or the specialization of different cells to perform specialized functions, is known as cyto-differentiation. Then, let us see what are the factors affecting cyto-differentiation. The first factor affecting cyto-differentiation is the phytohormones. Auxins at low concentration stimulates xylogenesis, and when auxin and kinetin are used together, they have a synergistic effect. Cytokinins and gibberellins also stimulate tertiary element differentiation. The second factor is sugar. Sucrose plays an important role in vascular tissue differentiation, and its concentration directly affects the relative amounts of xylem and phloem formed in callus tissues in the presence of low concentrations of auxin. 1% sucrose induces little xylem formation, while 2% sucrose induces better xylem differentiation with little or no phloem. But when sucrose level was increased to 2.5 to 3.5%, both xylem and phloem are differentiated. However, when sucrose concentration is further increased to 4%, phloem differentiation takes place with little or no xylem differentiation. The next factor is nitrogen. The presence of ammonia and nitrate in the medium directly affects the differentiation of tertiary elements. The last factor affecting cyto-differentiation are the physical factors. The effect of light on vascular tissue differentiation varies between cultured tissues, and temperature and pH of the medium also has effects on cyto-differentiation. The temperature within 17 to 31 degrees Celsius generally promotes vascular differentiation. Let us now take up morphogenesis in vitro. Morphogenesis is the attainment of biological organization or form, and morphogenesis in vitro is the reconstitution of an entire plant with similar shape, structure and function of the mother plant from cells or organs cultured in vitro. Under in vitro conditions, morphogenesis can be achieved by two routes. The first one is de novo origin of organs, either shoots or roots, from the cultured tissues, precisely termed as organogenesis, and de novo origin of embryos with distinct roots and shoot poles on opposite ends from the somatic cells or cells cultured in vitro, called as somatic embryogenesis. First, let us see what organogenesis is. In plant tissue culture, organogenesis is referred to as the development of organized structures such as shoots, roots, flower buds, somatic embryos, etc. from cultured cells or tissues. In organogenesis, the shoot or root may form first, depending upon the nature of growth hormones in the basal medium. In vitro plant tissues may also produce many types of primordia or adventitious buds and other organs, including those that will eventually differentiate into embryos, flowers, leaves, shoots and roots. These primordia originate de novo from a cellular de-differentiation process, followed by initiation of a series of events that results into an organ. Let us now see what are the events that takes place during organogenesis. Organ formation occurs through the process of differentiation in the callus or undifferentiated mass of parenchyma cells, which are highly vacuolated and with inconspicuous nuclei and cytoplasm, sometimes with lignification. In this group of cells, regions showing random cell division are present, forming regions of high mitotic activity, resulting in the formation of meristematic centers known as meristemoids. These meristemoids can occur directly on an explant or indirectly via callus. Normally, continued cell division in these meristemoids would produce small protuberances on the surface of the calli, giving nodular appearance to the tissues, which gives rise to primordia of organs that form either shoot or root. In organogenesis, the shoot or root may form first, depending upon the nature of growth hormones present in the basal medium. The genesis of shoot and root from the explants or calli is termed as collagenesis and rhizogenesis respectively, while the abnormal structures that develop during organogenesis are called organoids. Further, organogenesis may be of two types: direct organogenesis and indirect organogenesis. First, let us take up direct organogenesis. Direct organogenesis is accomplished directly from the explants without an intervening proliferative callus stage. Here, the primary explant gives rise to meristemoid, which further leads to the development of organ primordium. Direct organogenesis can occur depending on the hormonal combination of the medium and the physiological state of the explants. Although sucrose concentration and light modify the response, structures produced depend mainly on the auxin to cytokinin ratio. Medium supplemented with relatively high auxin concentration will promote root formation on the explants, and high cytokinin concentration will promote shoot differentiation. The effect of growth regulators on morphogenesis may be summed up as follows. High cytokinin to low auxin ratio leads to regeneration of shoot pod. Then, the presence of low cytokinin to high auxin leads to regeneration of root pod. Medium cytokinin with medium auxin leads to regeneration of both shoot and root. Or medium cytokinin with low auxin leads to callus formation. Then, direct organogenesis may lead to number 1, direct embryogenesis. It is the organized development where somatic embryos develop directly on the explants. Direct somatic embryogenesis proceeds from cells which were already embryogenically competent while they were part of the original differentiated tissue. The second is direct differentiation of organs, leading to formation of shoots or roots directly on the explants. The ability to undergo direct organogenesis may be linked to the developmental age of explant tissue, with cells progressively losing this potential as they mature. The next is indirect organogenesis. Indirect organogenesis involves an intervening callus stage which can later be induced to develop into organized structures. In indirect organogenesis, the primary explant gives rise to callus, which leads to the development of meristemoid, and the meristemoid further develops into organ primordium. Fully mature cells, if they retain any capacity for de-differentiation, tend to exhibit totipotency through indirect organogenesis. Auxin is essential for de-differentiation, leading to callus formation, and commonly 2,4-dichlorophenolic acetic acid or 2,4-D is used to promote callus, and cytokinin often enhances this process. Let us see somatic embryogenesis. This is another major pathway of regeneration and development of plantlets from plant cells or tissues cultured in vitro. When cells are cultured under a particular hormonal combination, the physiology of the cells change into a state similar to zygotes and follow an embryonic path of development to form somatic embryos. Somatic embryos are also called as embryoids, accessory embryos, adventitious embryos, or supernumerary embryos. And the process of somatic embryo development is called as somatic embryogenesis. Now coming to the conclusion. The regeneration of whole plants from plant cells or tissues cultured in vitro provides the most compelling evidence for cellular totipotency. For expression of totipotency in plant cell or tissue cultures, a differentiated cell first undergoes de-differentiation followed by redifferentiation to regenerate an entire plant from the cultured explants directly or via callus indirectly. Morphogenesis, or the reconstitution of an entire plant with similar shape, structure and function of the mother plant from cells or organs cultured in vitro, occurs either by organogenesis or de novo origin of organs like shoots and roots from the cultured tissues, or somatic embryogenesis or de novo origin of embryos with distinct root and shoot poles on opposite ends from the somatic cells or cells cultured in vitro. Thus, the regeneration of whole plants from cultured plant cells or tissues in vitro shows the expression of the phenomenon of cellular totipotency and in vitro morphogenesis by plant cells.